Can you see, children? One of you unmute yourself. Can you see the screen?
Yes, ma'am.
So you can see neural control and coordination, right? So we are beginning with the new chapter, that is control and coordination. That is related to what? Brain, spinal cord, and then different types of glands. So this is what we will be starting with, the nervous tissue, which includes neurons and glial cells. There are two types of cells in the nervous tissue which are considered to be the part of nervous system. So we'll start with the structure of neuron. This is what you have learned last year, but still don't know whether you remember everything or not. So this is the structure of a single neuron. Neuron is one type of cell, and glial cells or neuroglial cells, there are two types of cells in the nervous tissue which are considered to be the part of nervous system. Then we will be studying in detail. So nerve cells are of two types. Two types of cells in the nervous system: neurons, which receive and transmit the information to other cells. Around 100 billion to 1 trillion neurons are present. Okay, and the other type of cells, that means glial type of cells, or they are also called as neuroglial type of cells. These are considered to be the substituting or additional or helping cells. These are considered to be helping cells, which help the neuron cells. So their different their function is not to transmit, but they cannot transmit the information like neurons, but they help the work, action or functioning of the neurons, and their number is more than neurons, almost 9 times that of the neuron. So now we'll come to the neuron, that is the components of the neurons. That these are dendrites, soma, that is also called as cell body, axon, and presynaptic terminus. The main are soma, that means the cell body, and dendrites, different types. Now, if you see the structure in this diagram, you can see this part uh, is shown in this circle, right? So the entire structure is called as cell body, or this is also called as cyton. Remember these all words: neuron, cyton, dendron, or axon. These are different words related to the neuron, but what the main thing, the neuron is made up of cyton, that means this cell body, and different types of dendrons. So these are the dendrons. I'll explain in detail in the another diagram, and the axon. So basically, two parts are there. That is this part called as cell body or soma, and these extensions which you can see here, they are called as dendrites, and this cell body is nothing but the cyton, and these extensions are called as dendrons. And when they are again branched, they are considered to be known as dendrites. And one of the dendrons, it gets extended to form a axon. So the extension of the dendron, one of the dendrons gets extended, that is called as axon. Now try to understand here. Neurons consist of the main part called cell body, which contains the nucleus and various organelles. Means you can see here at the central place, the nucleus is present, and like remaining cells, if you remember the structure of cells, normal plant cell, animal cell. Now, animal cell, if you remember, along with the central part, that is nucleus, remaining molecules, organelles like ribosomes, mitochondria, Golgi complex are present, right? So here also in the cell body, that is the cyton part of neuron, at the center nucleus is present, and then in the cytoplasm of the neuron is called as neuroplasm. Just related to neuron, it is called as neuroplasm. Okay, we have learned it. Just this is the recollection, you can say. You can recollect with the help of this diagram again. Now you can see in this diagram that these extensions of the cell body here, which are shown, these are called as dendrites. Okay. So these single is dendron, branch called as dendrite. So these are all dendrites. And if you see, these neurons contain the cell body extensions. They are called as processes or neurites also, which carry impulses to and from the cell body. Now, what is towards the cell body, which are called as dendrites, and which are away from the cell body, they are called as axon. This is called as axon. That we'll see in the next diagram. So, two different types of processes can come from the cell body. The first is called as dendrite, all these extensions, the shorter ones, these all, and the extension, larger one, large extension, or the larger one is called as axon. Now, if you see the directions, then you will come to know, remember very easily, all these short extensions of the cell body, they are coming towards the cell body. These are the dendrites. They are carrying the impulses towards the cyton. Cyton is nothing but the cell body. Okay. So remember, they are also called as afferent processes. The afferent means towards. Afferent processes means nothing but these all extensions. These are called as extensions or processes. So these processes which are coming towards the cell body, they are carrying the impulses towards the cell body, that means cyton. They are called as afferent processes. Afferent means towards. And the extensions which are moving away from the cell body, that here the you can see the direction also, that the extension, this is the extension that is carrying the impulse away from the cyton. You can see the arrow, it is away from the cyton. So this is called as axon. So axons are nothing but the efferent processes. Processes is the extension. Efferent means away. Afferent means towards. Remember, afferent is towards the cell body, cyton. And efferent only is the difference of A and E. A, that is towards. E, efferent, that is away. So the extension which is moving away from the cell body or going away from the cell body, which is carrying the impulse from the cell body away, that is called as axon. Okay, so remember this direction. Now, in detail, very clearly you can see the remaining parts also, that is the cell body, that is also called as soma. Then this is the, if you see, these are the dendrites. Then this is the cell membrane. Cell membrane is also called as neurolemma. The another word is what? Neurolemma. Then here, when this starts extension, this region, that axon, where the axon starts, that is also called as axon hillock. That is a little bit bulging structure, which is not shown clearly here in this diagram, but that is called as axon hillock, which controls the information to be passed to the next nerve through axon, that is axon hillock. And whichever the diagram here, you can see here, these axon is surrounded by the sheath that is called as myelin sheath, which is considered to be insulating substance, that is made up of fats and some other substances which are helping to make this axon as an insulating substance, which helps to carry impulses in a faster rate. One more thing you can see here, that is, this is the gap where there is no myelin sheath. There is no sheath around this axon, that is called as node of Ranvier. This is called as node of Ranvier, where this part of axon is not covered by myelin sheath. And these myelinated nerve fibers, or this structure, that is also called as Schwann cells, that I'll explain in the next diagram. And one more, generally these axons are unbranched, but sometimes whenever they are showing branches, that is called as oligodendrites. Then this is called as oligodendron or oligodendrites. And these are the Schwann cells, or these are the myelinated sheaths which keep these, which are helpful for carrying the impulses in a faster way. That means from here it will jump here and then from here. So whenever you just see that when you walk and when you make a jump very fast, within few seconds that maximum distance is carried, right? So this way these electrical impulses are transferred in a faster way with the help of this tree. And here the extension, the last part of this axon is again, if you can see the branches over here, that is that it when this axon is terminating, that means the end of the axon, that also shows these branches. So these are called as telodendrites. These are called as telodendrites. And the end points of these telodendrites, their synapse. Synapse means the simple word. The meaning of the synapse is what? Pairing. Synapse is what? Pairing. Pairing of what? Pairing of the dendrites of the another neighboring cyton, or you can say neuron. So here at this point, pairing of the another neuron takes place with the dendrites of the another neuron. Telodendrites of the this neuron takes place, and that point is called as synapse or synaptical complex or synapses. That you can consider it as synapses. Okay. Now, here one by one in detail, little bit in detail, we'll go about the dendrites. These are branching fibers with a surface line with synaptic receptors responsible for bringing information into the neuron. I showed you that here, whatever these branches, these dendrites, here also they are having the points, end points, okay, which fuse with or synapse with or pair with the neighboring neuron, that is telodendron part of the neighboring neuron, okay, which I showed just now. So bringing information into the neuron. Some dendrites also contain dendritic spines which further branch out and increase the surface area of the dendrite. Just now these, which whatever the more branches I showed you, these are nothing but which looks like spines, right? More branches of the main single is dendron, then gets branched into dendrites, and then again small branches which help in increasing the surface area to get maximum information easily, fast, in a faster way. So they are called as spines. Dendritic spines are the points of communication between two neurons. Now the next is what? Cell body, as I showed you just now, contains the nucleus, mitochondria, ribosomes, and other structures found in other cells. Also responsible for the metabolic work of the neuron. Different type of metabolic activities are performed with the help of different organelles present in the cyton, that is the cell body. Now, about axon, that is a thin fiber of a neuron responsible for transmitting nerve impulses towards other neurons, organs, or muscles. Single axon extends out of an expansion of the cell body known as axon hillock. Just now I was telling you what is that axon hillock. When that axon starts its extension and gets little bit expanded, that expanded portion of that axon very near to the extension where it starts, that is called as axon hillock. Axon may have branches called axon collaterals. I showed you previous branch. Generally these are unbranched, but whenever the branch is seen, that is called as axon collateral. Laterally it is present, parallel to that it works, that is what collaterals. Axons covered with an insulating material called myelin sheath with interruptions in the sheath known as nodes of Ranvier. I showed you in the diagram, right? So axon collateral divides into small branches called telodendria. You know, just now I showed the end of the axon where it gets terminated. That part shows branches. These small branches are called as telodendrites or telodendria. And knob called end fruit or terminal button at telodendron. That is that particular knob where it ends, that is telodendron. And from there the branches starts, that is telodendrites or telodendria. Then presynaptic terminals refer to the endpoints of an axon where the release of chemicals to communicate with other neurons occur. I showed you that synaptical terminal also in the diagram. Now here again you can see this is the axon terminal. Okay, this is the cyton. These are the neurofibrils. These one more thing I forgot to tell you, which is very important part of the cyton, that is called as Neissel's substances or Neissel's granules. That means you can see these yellow colored bodies. These are called as Neissel's granules. They are nothing but the part of ribosomes. Means they are produced from the ribosomes, and that means the site. What is the main function of ribosomes? They are made for the process of protein synthesis. Ribosomes are the part of protein synthesis. So these Neissel's granules, they are helpful in the synthesis and then storage of proteins, which help in the nourishment to that neuron body, that is the cyton. Okay, so these are the Neissel's and these are called as neurofibrils, and this extended part that is called as axon hillock. Just now I was telling you from where the extension starts, the very entry from where it starts, that is called as axon hillock. Okay, and if you see the direction, see the neuron from the axon, it is coming this way. This is the collateral branch which is showing the branch of this axon, and the information is carried. It is jumping from one Schwann cell to the another, that is singular one is called as Schwann cell, and the gap between the are these neighboring Schwann cells is called as node of Ranvier. So Schwann cells forming the myelin sheath on axon, and when you see the direction how the nerve impulse is moving very fast with the help of Schwann cells, here is the end. Here this is called as telodendron, that end point, and from there again it shows the branches, telodendrites. This is the terminal end of the axon, and this is the point called as axon terminal, where this branch here, if this point is responsible, these points are responsible for synapses. That means the dendrites of the neighboring neuron get synapsed or pair with these points to get the information from the neighboring dendron. So this is very clear, I think, in this diagram. Now here again, with the help of this diagram, you can see how this works. Information flow through the neuron. See, dendrites collect electrical signals. See the direction here. They collect electrical signals, and from the neighboring neuron, that is the telodendron of the neuron, and then through dendrites, it is passed to the cell body, that is the cyton, and then from here through the axon, here what will happen? It integrates incoming signals and generates outgoing signals to the axon. Now outgoing signals are going through away with the help of axon and passes the electrical signal to the dendrites of the another cell or to an effector cell. Now what is effector and what is the sensor, that I will explain you in the next part. So these are the telodendrons. This is the end point, and here again these are the points of synapses. Synaptimal complex. This is also called a synaptimal complex. So dendrites to cyton, that is cell body, and from cell body to axon, and again from axon through telodendrites to the neighboring dendrites of the neuron. This is the way. Dendrites and axon are simply fluid-filled extensions of cell body. Remember, both dendrites and axon are simply fluid-filled extensions of the cell body. Information flows from the dendrites to the cell body and then axon. Dendrites receive thousands of data, but at single axon, data passes will be an average level and summarized version, and it is regulated by axon hillock. I just told you the function of axon hillock, because the number of dendrites is more, so the collecting information is more, but then when it is to be passed, then with a single axon, that information is passed in an average way, where that is summarized there, and then it is passed, and that means information is regulated over there at the axon hillock, which is to be passed first or next, like that. A neuron consists of a flow of electrical current that begins on the dendrites and then travels along the axon to the terminals. Each impulse reaches an end foot. The end foot releases a chemical into its synapse, that means that part, and the chemical influences the electrical activity of the receiving cell, thus passes the information. The chemical is known as neurotransmitter. Transmitter is what which is to be transferred, and that chemical which is present in the nerve cell, that is a neuron, that is called as neuro. That's why this is the name, that is neurotransmitter. Like other cells, neuron cell contain the following structures as I told you, that is the membrane, nucleus, Golgi complex or Golgi body, mitochondria, ribosomes, endoplasmic reticulum, then smooth and rough ER, all the organelles which are generally present in the normal eukaryotic cell, animal cell, both are present. And this is just the extended diagram where you can see this is the cyton and this is the centralmost part. This entire structure, yellow colored structure, is nucleus. Inside the nucleus, nucleolus is present, and you can see these extensions of the nucleus. This is the nuclear membrane, and the extensions of the nuclear membrane get converted into ER, then smooth and rough ER, then mitochondria, Golgi complex, ribosomes, polyribosomes. So all these remaining organelles also present. This is again clear, more clear here in this diagram. So you can see this. This is nothing but the cyton. So neuron consists of cyton, that is the cell body, dendrons or dendrites, extensions, smaller extensions, and axons. Axon, that is a larger extension. Okay. Now we'll see another type of cells, that is neuroglial cells, or they are also called as glial cells. Simply they are called as glial cells. They are considered to be the substitute cells of the neuron. That means what? They are helping cells, assistant cells, you can say. They are helping cells of the neuron. So the main function of the neuron is to collect and transmit the information. Here they help the neuron but can't transmit the information. Now there are different types of neuroglial cells. Some of these we will discuss here. Oligodendrites, as I showed you just now in the diagram, these are myelin secreting cells of the central nervous system. Try to understand central nervous system. Okay. Then astrocytes, I will show you the diagrams also later. Astra is related to the star, means star shaped cells. These are which provide physical and metabolic support to the nerve cells. Microglia, micro means small in size, or they are also called as microglial cells. They are also called as mesoglia, which are phagocytes of the CNS, central nervous system. You know the meaning of phagocytosis, cytosis, phagocytosis, pinocytosis, I have explained previously, but still, if you remember when we were studying the WBCs, monocytes and all, that time that word has come. So what is the role of phagocytes? They engulf the foreign material or the organism. So a little bit few more points we'll add about the microglia, means the role. These are, as the word suggests, micro is smaller, very smaller, smallest of the all glial cells. These are smallest of the all glial type of cells with short twisted processes. They are also having the processes like the neuron, that is the dendrites. They are phagocytes of CNS, considered part of mononuclear phagocytic system. Mononuclear is what? A single nucleus is present. If you remember monocyte, mono is single, that's having the single nucleus, but what is the role? That is phagocytosis. That whenever the foreign material is entered, that they engulf these foreign bodies or whatever the material comes, and there it gets digested by these cells. So they are helpful in immunity, as well as you can consider it as a defensive mechanism. They are defensive in function. They are believed to originate in bone marrow and enter the CNS from the blood. So originated at bone marrow, but then they enter CNS through blood. In the adult CNS, they are present only in small numbers. and in adult their number is less, but proliferate and become actively phagocytic in disease and injury. But whenever there is some disease, that person is affected with some disease or with the injury, automatically their number gets increased. You know why? Because they are defensive in function mechanism. Because whatever the foreign material comes by the injury, there are more chances when there is entry of some other microorganism or any disease causing organism, or through injury any foreign particles or microbes enter that, at that time that the system gets protected with the help of these microglia. So their alternate name mesoglia reflects their embryonic origin from mesoderm. So why they are called as mesoglia? Because their origin is from mesoderm. And the rest of the nervous system, including the other glial cells, is of neuroelectrodermal or neural crest origin. Just remember. Now, one more type of cell, that is the support cells of PNS, called as satellite cells and Schwann cells. Satellite, they are they surround the cell bodies, means cyton, of the neurons in ganglia. Gangliated regions are there. When we'll study in detail this peripheral nervous system and all, that time we'll come to know. So there on the dorsal side of the ganglionated region, these satellite cells are present around the cell body, means they cover the cell bodies to protect. Okay, the cell bodies of the neurons, that means cyton. These small cuboidal cells form a complete layer around the nerve cell body, that means cyton, but only their nuclei are visible in routine preparations. We can see only the nuclei of these satellite cells. They help to maintain controlled microenvironment around the nerve cell body, that is cyton, providing electrical insulation and a pathway for metabolic exchange. See, like axon is having insulating substance as myelin, here these satellite cells surround this cyton and provide electrical insulation and also a pathway for metabolic exchange. Now, Schwann cells, as I showed you, that these are responsible for myelination, the axon, the PNS, and Schwann cell wraps itself jelly roll fashion in a spiral round a short segment of an axon. Means at a particular distance they wrap, again there is gap, then again the wrapping is there. That is what called as, during the wrapping, cytoplasm is squeezed out of the Schwann cells, and the leaflets of plasma membrane of the concentric layers of the Schwann cell fuse, forming the layers of myelin sheath. So in this way, by wrapping, it forms the sheath around itself at axon. Now these are the diagrams where you can see different types of glial cells or neuroglial cells, which I showed you this oligodendrites with the extension, parallel collateral extension of the axon, that is oligodendrites. Then ependymal cells are nothing but the epidermal cells which originate from the columnar epithelium or squamous epithelium, or sometimes ciliated epithelium of the central nervous system, that is a part of the ventricles of the brain. That we will come to that point when I will come to that point, that I'll explain again. And this is what the satellite cell, where the nucleus only is prominent, just now we were discussing. Then this astrocyte, that is the star shaped, which gives the appearance of aster, that means star shaped structure. Then these are the microglia, very small in size, and that with the single nucleus at the center, and monocytic, that is the phagocytic in function, and this is the Schwann cells, where axon is covered by this myelin sheath. So these are different types of neuroglial cells. Now, morphologically, this neuron is divided into three categories, types, that is unipolar, multipolar, bipolar. Now we'll see how morphologically it is different, means appearance. Morphological classification means how it looks, how it appears. Now the word itself suggests uni, single, multi is many, bi means two, right? So we'll see one by one, step by step. Unipolar neuron. Now if you see here in this diagram, this is the cell body cyton, that is the single cyton. It is having the only one extension. This is what cell body, and then it is showing these extensions called dendrite. This is one dendrite of the cyton, and this is the axon. So has only one pole, that is what called as unipolar. Unipolar neurons, that is the cell body, has a single nuclide that divides a short distance from the cell body into two branches, one proceeding to some peripheral structure and the other entering the central nervous system. And examples of unipolar neurons are, they are found in the posterior root of ganglion. Then that, what is that posterior root? That is when we will study the next part of the spinal cord and all. That time I will explain you. Just remember that this is the morphologically only single pole here. One extension is moving away called as axon, and the one extension is which is coming towards, that is called as dendron. So dendron and axon, only one pole, so unipolar. Now the next, that is bipolar. Okay. Now here you can see bipolar neurons possess an elongated cell body. The structure is different. You can see there from the cell body, the branches were there. From here directly, the structure is like this. From there itself, it is showing one, that is the shape elongated cell body, from each end of which a single neurite emerges. Examples of this type of neurons are found in the retinal bipolar cells and the cells of the sensory cochlear and vestibular ganglia. Can you imagine what does it mean? If you remember the structure of eye, and if you have learned to some extent in the previous classes the structure of eye, retina of eye, even though you can see the different types of cells, rods and cones, if you remember. So there, this is what the example bipolar retinal bipolar cells are of this type. Bipolar neurons, they are of bipolar type retinal cells. And the another one, that the sensory cochlear and vestibular ganglia, that is what, if you remember cochlea, the word cochlea, where it is related to, obviously the word is here to help you remember and find out, or recollect, that is sensory means what? Sense organ only. What is that? Cochlea. Have you heard the cochlear transplantation and all? That is related to auditory nerve. That means what? Auditory means ear. This is what? Ear. So in eyes and ears, bipolar type of neurons are present at a particular place that is mentioned. Now, multipolar neurons, if you see whatever the structure we have learned so far right now, this is what we have learned, that is multipolar neurons have a number of neurites arising from the cell body. With the exception of the long process, the axon, the remainder of the neurites are dendrites, as we have already discussed. That this is only the extension, larger extension as axon, and the remaining are dendrites. So most neurons of the brain and spinal cord are of this type. So multipolar neurons are present in brain and spinal cord. So remember that. Now, according to the function, neurons are classified as sensory, which are also called as afferent neurons, motor, also called as afferent neurons, and interneurons or association neurons, or this is also called as mixed type of neurons. Okay, so we will see now what are these. Sensory, you remember, sensory, the word itself suggests that related to sense organ. You know, there are different types of sense organs, 5 main sense organs, you know that, right? So periphery of the body to the central nervous system, how they carry the information, and also called as afferent neurons. Afferent means what? Towards. Towards what? Neuron means what? Cyton. Okay, and you know what are these extensions from dendrites? Or whatever the extensions, from where? From sensory organ to the central nervous system. That is that particular part, that is here in this case, here, that is the cell body. From where it is information getting, from where it is coming? From the sensory organ. Suppose this is the skin. So these are the receptors. From the receptors of this sense organ skin, the information is carried to the neuron, that is the cyton, that is means what? Afferent neuron. They are called as sensory. So these neurons are usually concentrated in areas called ganglia, and their dendrite branches extend to the skin or two sensory organs and act as sensory receptors, either directly or indirectly. Okay, that means skin is one of the sense organ. Then remaining eyes, ears. Then what else? Taste, that is tongue. Then what else remaining? So all these five sense organs there, these extensions of the neuron are uh, present, and these areas in that particular skin, along with the skin remaining sense organs, these extensions are present, and these are called as receptors. These extensions of the neurons are called as receptors. So from there the information from the sense organ is carried towards the central nervous system, means to the main body. Okay, so these are called as afferent neurons. Now another one, that is motor neurons. Motor, the word itself suggests that moving. Motor is what? In the moving state, right? So motor neurons are also called as efferent. As I told you, efferent, that is away. These neurons carry impulses away from the cell body, that is the central nervous system, and thus the central nervous system to muscles, gland, or some other effector to produce a certain action. So for these motor neurons, they, whatever the information is received by the afferent neurons to the central nervous system, that central nervous system, the brain or spinal cord or whatever the cell body of any of this will uh, transfer this information into the effect, and so they are called as efferent. Efferent means they are moving away from the central nervous system to the effector, means wherever that effect is to be shown, so to produce certain action. Most of the neurons in the spinal cord and many of those in the brain are motor neurons. Remember this. So because from the central nervous system, that is maybe from the brain or the spinal cord, the message is given to that effective effector. That means that particular organ. That means whatever the action is to be done. So they carry the information away from the central nervous system. So they are called as efferent neurons. And as they are carrying the information is moving, that is, these are called as motor neurons. Interneurons or association neurons, that is the third category, that is considered to be the combination of these two, or connector, you can say. This type of neuron is restricted to the central nervous system. They are also called connector neurons. These neurons act as bridges between sensory and motor neurons. They are considered to be the bridges between the sensory and motor neurons, or relay impulses to various functional centers of the brain or spinal cord. Okay, so this is what the classification according to the function, as well as classification according to the morphology. So these three types, we categories we studied, right? Now the actual part of the main system, that is the human central nervous system or neural system, we will come here and we'll discuss in a few more minutes are there remaining. So uh, this is what we'll start with the brain. As you know that the central nervous system, which shows the presence of brain and spinal cord. So let us study about brain, what type of organ it is, whether it is busiest
Hello, hello. Kotwal madam, can't hear you.
and
Kotwal madam,
protected.
voice is breaking.
Yes, uh, same device.
Hello. Students, please wait. I think there is some internet problem as she had told earlier. I'll call her. Please wait. Hello. हाँ तो प्रेझेंटेशन मधनं बाहेर पडलीये का? नाहीये. नाही. आवाज पण गेला होता मगाशी. आता पण नाही येते. नाही. नाही प्रेझेंटीचं चार्ट आलाय. हा. It's already on the main screen.
Now.
Now audible, but presentation गेलाय.
Now audible?
Yes, but presentation can't see, yeah?
No, I have come out of that. So I will start again. Okay.
Okay.
Can you hear me now? Clearly?
Yes. Yes.
Just 15 minutes we will continue. Not more than that. Okay.
Okay. Hello? अगं पण मला शनिवारी इंग्लिश आणि IT दोन्ही दिसतंय बघ टाइमटेबल.
Okay, can you see me now? Sir, can you see the screen now? One of you just unmute and just let me. You can see the screen now. Okay. The previous few slides, I think you couldn't see. I will repeat those slides. Okay. Did you see this or no?
No, ma'am.
Okay. Just a minute. Now we come to the human central neural system. That is which includes brain and heart. Start one by one with brain. So let's study about brain, which is considered to be the busiest organ. Okay. Last time we studied the very important busy organ as heart, and now we are going with this next one at brain. Brain is also called as encephalon. N means in, cephalon means head. So it is present in head. Now can you hear me? Please one of you unmute. Is it clear now?
Yes, ma'am.
Yes, ma'am.
And you can see also visible?
Yes.
Oh. See, in the box, that brain is protected, covered by a box that is called as cranium. So that's why if you remember the word craniata, that means all organisms which are having this cranium, that is the brain box, are included under craniata. That means cranium is nothing but the brain box, that is bony brain box. Okay. So in this way it is protected. Now if you see, average weight of the adult human brain is 1200 to 1400 grams, but which brings it down to just feeling of 20 grams in head. You know why? Because of the fluid which is present in the brain, that is called as CSF, that means cerebrospinal fluid. So because of the presence of cerebrospinal fluid, the brain is floating in it. So because of the fluid present in it, that is CSF, that weighs, we feel only of 20 grams, but actually it is 1200 to 1400 grams. Now this is the actual what we have to start learning, the part that is about the nervous system. See, you can see here these three parts. First, central nervous system, peripheral nervous system, and autonomous nervous system. First, central nervous system. Second is peripheral nervous system. Third is autonomous nervous system. We'll start with the central nervous system. Central nervous system includes brain and spinal cord. Peripheral nervous system includes cranial nerves and spinal nerves, whereas autonomous nervous system includes sympathetic and parasympathetic nerves divisions. Now, when we start with the central nervous system, and in that first, that is the brain, which is also called as encephalon. Just we studied. N is in, cephalon is brain, the head. So in the head it is present. So encephalon. Now it is again divided into three parts: forebrain, midbrain, and hindbrain. So we are going to start with the first part, that is the brain. In that, which is divided into front part as forebrain, middle part as midbrain, and the behind back part or the last part is called as hindbrain. So forebrain is called as prosencephalon, midbrain is called as mesencephalon, whereas hindbrain is called as rhombencephalon. So these are the biological terms. You also should remember these terms. Though you know forebrain, then word also you should remember, that is prosencephalon. Okay, now when we start with the forebrain, again these are the three important parts of the forebrain, which is divided into olfactory lobes, then cerebrum, and diencephalon. So we are going to start with this first, that is prosencephalon. We'll see this is what in general the structure of brain. Now, this is what again the division of the nervous system shown in the diagram: CNS, TNS, and ANS. CNS includes brain and spinal cord. TNS includes cranial and spinal nerves. ANS include that is functioning with the control of internal organs. Okay, and this is the uh, neuroectoderm. Ecto is outer, so that develops from neuroectoderm, from the neural nervous system, which is related to the nervous, and ecto is the outer. So this is the neuroectoderm, outer layer, and this is what this part as brain and this part as spinal cord. That is what CNS includes, brain and spinal cord. Now, brain is considered to be the living supercomputer. You know, everybody knows computer. So the information is collected here. So here it is the living supercomputer. So the brain is the site of information, processing, and control. So information is stored, processed, controlled, everything. Now, when we study the structure, meninges means the covering. Meninges means layers or coverings, you can say. Okay, how this brain shows the different coverings, because it should be protected, because it is very delicate and the most important part of the body, isn't it? So that's why it should be protected. So these are the protective layers called as meninges. So these layers or coverings are called as meninges. So there are mainly basically three layers. These meninges are of three types. So what are these? In this case, you can see the structure. The outermost, that is called as dura mater, which is associated or attached outwardly to the cranium. Then the second membrane, that is called as arachnoid matter, and the innermost layer, that is called as pia mater. Now this is what outermost dura mater, the middle one arachnoid, and the inner one, that is pia mater. So the brain is divided into these three parts. The dura mater means this is the outermost, tough, thick, and fibrous, which is attached to the inner side of the cranium, and the main function is what? Protection. So dura mater protects the inner parts like arachnoid and pia mater, and inner parts, inner layers, and all the inner parts of the brain. So protective in function, which is the outermost tough and attached to the inner side of the cranium. Now, arachnoid matter. This is the middle one. Arachnoid matter, this, here you can see this is the dura mater, this is the part of cranium, and this is the arachnoid matter. Here also the branches or extensions are there called the trabeculae. That means these are the extensions like structure, they are present. This is the middle part, middle layer of the brain, that is called as arachnoid matter, and the innermost called as pia mater. Okay, this is the innermost. So dura, arachnoid, and pia mater. These are the three different layers. They are called. They are considered to be the part of the meninges. Okay. So pia mater, that is the innermost, thin, contact with CNS, that is central nervous system, highly vascular in function and nutritive in function also. This is highly vascular, which is helpful. This and it is the nutritive in function. Now, sub means below, and serous means fluid, which I was just mentioning about the CSF. That is, here if you see, this is the uh, part outer layer. This is the dura mater. Here, the fluid is present between the two membranes, dura mater and arachnoid matter. Here, the whatever the fluid is present, that is called a serous fluid, serum or the fluid. That is what serous fluid. That is what CSF. Okay. Again, between the arachnoid and pia mater, wherever it is present here, that is what called as here it is subdural fluid. Sub means below the dura mater, that is called as subdural fluid. That means between the dura and arachnoid, whatever the space is present, that is called as subdural space, which is filled by fluid called as CSF, cerebrospinal fluid. So in this region again it is shown, this is what dura mater, this is arachnoid matter, and this is pia mater. So this is filled in between these two layers, whatever the fluid is present, that is called as CSF, that means cerebrospinal fluid. Here again it is very clearly shown. Here this is the region where cerebrospinal fluid is present, and the different meningitis means what, you know, everyone know, that is brain fever, or that is called as meningitis fever in brain. If you have heard this word meningitis, because of the infection to this brain, infected cerebral fluid, spinal fluid, cerebrospinal fluid, it gets, this tissue gets swollen, that the tissue in between these layers that gets swollen, and so that is what called as meningitis. So this cerebrospinal fluid is present between these two layers. Remember, this is the normal meninges. Now, what is the function of CSF? It is working as shock absorber, constant pressure. The pressure is maintained. Pressure of these matters or these layers or whatever the internal organs of the brain, that is maintained. Then it regulates the temperature. Then exchange of nutrients, that is also very very important function of the CSF. Then exchange of wastes. Then supply of oxygen, that is very important, and protects the brain from desiccation. As it is present there, that protects the brain from desiccation. So this is the human brain or encephalon. So functions of the nervous system: controls and coordinates, rapid communication, constantly aware, controls voluntary activities as well as involuntary activities, and then thinking, reasoning, memory, etc. These are the functions of central nervous system. And the characteristics of brain, that is, it is soft, whitish, large sized, slightly flattened, 1300 to 1400 grams in weight, and the volume is about 1300 to 1500 cc, and gets developed to full size at the age of 6 years, and contain 100 billion neurons. Remember, that contain 100 billion neurons. Now, parts of the brain and all, I think we will discuss next time. These are just parts of the brain. I will just mention you that the parts, that is the forebrain, front one, midbrain. When you see the LS of brain, you can see these parts like this, parts of the brain. We will discuss it detail next time, starting with the forebrain. Forebrain, the front one, midbrain, the middle one, and the hindbrain, that is the back side of the brain, back part of the brain. So forebrain is called as prosencephalon, midbrain as mesencephalon, hindbrain as rhombencephalon. Okay, so I will stop here, and whatever the doubts if you have, you can ask me. I stop my recording and presentation also.